I'd like to welcome the next speaker, Maduria. Um, I, I came to know her from 2016 when she came into the UIC in the OTM. Her background, she has completed her PhD in chemistry from Texas A&M. In 2000, she continued from there to the University of Minnesota for a postdoctoral fellowship in biochemistry and molecular biology. Then she moved to NCI, National Cancer Institute, for a second postdoctoral fellowship in the laboratory medicinal chemistry. Now it's called the Chemical Biology Laboratory. But for some reason, she didn't like to stay in the lab. She decided to transition to a career in technology transfer. So within the NCI, there's a technology transfer center for the National Cancer Institute. So in 2005, she became a technology transfer specialist over there. So while at NCI, she promoted technologies that developed at the NIH, attract industry partners to further develop the commercialization, the technologies to benefit the public health. And then following that, she completed a master's in management with marketing in 2011. And subsequently, she finished an MBA in 2013 at the University of Maryland. And that's when she was looking for universities in the US where there are smart scientists working. She found UIC. So she joined the UIC Office of Technology Management in 2016 as the assistant director and subsequently became the senior technology manager. At that time, she was responsible for managing the portfolio from the College of Medicine, including all the evaluations, patenting, marketing, and the licensing activities. She worked with the College of Medicine faculty closely and promoted UIC technologies to attract industry partners, of course. And I was one of the beneficiaries. I can gladly say that she was totally responsible for marketing the dog vaccine. And she went into one of those meetings and promoted that over there. So that has led to the current marketing of our current technology transfer at this point with the Merck. She also promoted UIC technologies to attract industry partners for further development and commercialization. So currently, Melissa is the scientific collaboration director with the West Loop Innovations, a partnership between UIC and Deerfield Management to accelerate the commercialization of therapeutics at UIC. So Deerfield has set up to $65 million in translational research. She's going to talk about that, mainly to provide funding for the state-of-the-art drug development. So as the scientific director, Melissa manages all aspects of the West Loop, including engagement with faculty, solicitation of proposals, overseeing review process, and also works with the UIC faculty with the broader Deerfield drug discovery development. Melissa works with the principal investigators to develop research plans and allocate the resources needed to achieve the IND readiness and the first step towards the clinical testing. So, Melissa.
Jen said I'm supposed to change my name. Let's see if I can do that. I don't think that's going to happen. We're going to leave that alone. Okay. So I'm Melissa Maderia. So I'm going to tell you a little bit about what I've been doing with West Loop Innovations. And I'm hoping I can step over here so that I can see everybody. They can still hear me on the mic. And then I also want to tell you a little bit about some of the insights that I've learned while I've been in this role, working with Deerfield Management, who's a venture capital firm with $1 billion capital, in terms of what I've learned in terms of drug discovery insights. So what I've learned that can help researchers. So when you're in the early stage of research, you don't often think about what do I need to think about for those later stages in order to take what I'm developing in my research in my lab to develop a drug. And so some of the slides are going to have a lot of information on them. Dahlia has copies of these slides. You're welcome to get them from her, or I can send them around to Ramaswamy and Matthew, and you guys can distribute them. There's a lot of information in these slides. I'm going to go kind of quickly through some of it. Then I'm really happy to talk to you guys about what you're doing in your labs and to learn how we can either potentially partner with West Loop, or how I can just help you generally in your lab and working with OTM. And I work really closely with Eugene and Nelson. So almost any meeting that you have me in, you'll have one of the two of them and also. So West Loop Innovations is a partnership between UIC and Deerfield. Back in 2018, we signed this contract where Deerfield basically pledged $65 million to UIC in order to fund roughly about 10 projects at $5 million apiece. As these have been developing at other universities, we're finding that there have been closer to $15 to $20 million invested in these projects. So their idea is when they find something that's got a nice target, a nice indication, something they think they can get to market, they want to do everything that they need to do to get that into clinical trials. And so my job is really to be out here talking to faculty, finding out what you're doing in your lab, and putting those proposals in front of Deerfield so that we can get a therapeutic project funded and to the market. So what Deerfield identified back in 2015, 2016 was that there was a lot of challenges working with in an academic environment in terms of how to do drug discovery and how to take an early stage asset and move it all the way to IND ready. It was a very difficult funding environment at that time for basic research, and it probably still really is. NIH funding has continued to decline from the number of faculty that are coming on board at universities. Pharma and biotech investments were moving further and further out away from early stage research. And a lot of people were writing what are called safe grant applications. So focusing on mechanism, focusing on understanding science, but not really focusing on therapeutics and drug discovery. So they wanted to kind of fill that void. And so the way that they've done this and the way that they've addressed these challenges is really to leverage the people and the assets that Deerfield has in the life sciences in terms of their expertise and their operational capabilities. And so what they wanted to do is go out and identify really novel early stage technologies, novel targets, novel mechanisms, ways to approach a disease that hasn't already had any therapeutic in it. So they want a huge unmet need, and kind of figure out how they can help advance those sciences and get more therapeutics to the market. And so really their idea was to fund projects that advance compounds, and now they've moved into biologics and gene therapy as well. So an IND ready stage in a really collaborative, cost-effective manner, and to do it really quickly. So as you know, most of the time when you do have something in the lab here that might be moved into drug discovery, i.e. Shingrix. We saw Shingrix come out in the mid 90s, yes. And it took until 2019 to get on the market. Why? Well, that one actually moved pretty quickly into pharma and into the clinics really soon. It took about 7 years even for that to move into the clinic. And then it took 14 years of clinical trials because it's a vaccine. So it was a long history and development timeline, and it stalled about 3 times in that. So what their hope is that they can take that normal 7 years that it takes, and usually it's 7 to 10 years to take something from the time you have identified your lead molecule to get into the clinic, to take that and shrink that timeline. So they're looking to do it quicker. And so the way that they're helping to kind of bridge this value of depth with faculty is to basically fund money back into basic research. So by the time you get to the FDA, the more you understand about the mechanisms, the more you understand about what's going on with that molecule, the better you can answer the FDA's questions, the more likely they are to approve it. But they also then can help you with target selection and validation. They can use their network of operational capabilities and CROs in their network to do some SAR around the small molecules or even around the biologics. They can help with lead selection and optimization, with manufacturing, with doing the tox studies, and all of the IND-enabling work that it takes to get something to the clinic. And so really the idea is that they're bridging that valley of death between the basic research and the product development. So how do you go from the basic research that you guys have in your lab to applying that research, which academic researchers start to do, but you almost never get to the product development stage. At that point, it has to be spun out into a company or go to a big pharma. So they're looking to figure out how we can do this in an academic environment, partnering with basically a virtual company, which is the Deerfield. And so the goal of West Loop really is to advance multiple therapeutic projects to IND ready stage. It's a really simple concept. Find good science and get it to the market. Sounds really simple. Not so simple. The way that this process starts is a request for proposals. When we kicked off West Loop back in 2019, in November, we sent out a request for proposal. We got about 39 proposals in across UIC. From that, it moved through kind of Deerfield's process. They did a evaluation and selected some projects, and then they moved them to fully development plan. So we took 2 projects from that, and I'll talk a little bit about what we did there. We moved them all the way up to full proposals, mapped out all the studies that needed to be done from the early stage all the way to ready to present it to the FDA for an IND study. They were getting approved for funding, and the faculty left UIC. So that was my first year on this project, and so a little disappointed. But now at that point, we'd also talked to a lot of faculty and have been since kind of building that network back up and pitched a lot of other projects to Deerfield. And while they haven't funded any projects yet, from working with them and the insights we've learned, we've gone out and been able to take that, give advice back to the faculty, do 1 or 2 key studies, and instead of it going into West Loop, we've actually found licensing partners for 5 of those projects. So it's still been a very advantageous relationship with Deerfield in terms of what we learned and how we've been able to help other faculty, even though it hasn't had any projects yet funded for this. But we're still hopeful. Right, June? And the idea is at the end that there would lead to either licensing with Deerfield, or Deerfield would say, no, we don't want to do a spin out, but we think this is already interesting to pharma. We can identify a pharma partner and do direct licensing. And then Deerfield's interest in this is that they share with the university. So that 40% that comes back to the university, we share that 40% back with them on the back end. And if you saw June's numbers in the beginning, that revenue number that was $55 million last year and $43 million this past year, 95% of that is therapeutics. So we know that when projects come out of this and they're sharing in the back end, they're going to recoup that $20 million in the first year if we get a therapeutic on the market. So they're very interested in the success of this program and identifying the right therapeutics. And they're happy to share on that back end and putting money up front. So some of the advantages of the West Loop model is to the investigator, it's an opt-in process. So as you saw with the one faculty, we can go all the way up to full proposal writing with them. And when you get all the way, you would know exactly what you would need to do in your lab to get all the way to an IND and go, yeah, I don't think I'm going to do that anymore. That hasn't happened at any of the universities, but it could.
So how much investment has gone?
But at the same time, if you're going to do that and you've got $20 million sitting on the table as a faculty, I might wonder. But again, I think that it's a great way, even like I said, it's opt-in for faculty, but what we've learned is we get a lot of insights just from the conversations we have with the people at Deerfield. They're all 25, 30 plus year pharma people that sit in all these specialty areas of drug development and discovery. And so the insights we gain from them are just valuable in terms of understanding what kind of science we need to do to move it into a drug development candidate. The investigator retains an active role in the research. So you get to continue doing what you want to do in your labs if you come into one of these projects. And more importantly, you get some funding to do that work, or you get additional data that allows you to write for additional R01s and have stronger R01 submissions. So I think it's a great tool for investigators. Again, like I said, you get the sponsored research and you get access to a team of experienced drug discovery people, including writing business plans, market intelligence, biostatistics, epidemiology. And so I really tapped into a lot of that in terms of helping faculty develop other proposals. And so while I'm working with faculty to pitch for West Loop to Deerfield, I'm also working with Hyunjin and with Nelson as we develop these proposals. And when Pfizer comes in and says, hey, what science do you have in this area? Well, now we've got this nice proposal packaged up for West Loop that we're putting in front of Pfizer, we're putting in front of AbbVie, we're putting in front of GSK. So we're not putting all of our eggs in this basket, but this is a great way to get things started and really use me in this role to develop that that package that we need to put in front of everybody else. So currently there is actually 20 universities that are partnering with Deerfield. This is 18 of them on here as of last year. At Stanford's come on board and one other university the past year. So I tap in also to all these universities in doing this in terms of what they're looking at, how they're building projects and the expertise they have. And as you can see, we're looking at a lot of the world-class universities that are partnering with Deerfield. So it's kind of a nice reputation boost for UIC to be in the same conversation as Stanford and Harvard and Columbia.
So they all kind of go for the temple.
So they're all applying. So Deerfield has actually put 65 million in each of these schools. So it's, like I said, they've pledged over $1 billion basically in research between all of the universities across the US. So, but it's nice that UIC is thought of as a place that the type of research comes out of that Deerfield wants to, that we know we're in competition with the Ivy League and the other big schools. And so now I'm kind of transition a little bit and give you some of the insights about what I've learned in this role and how you can even apply this just in terms of your academic research if what you're doing is ultimately going to be leading to a potential therapeutic. And so really the way we think about drug discovery and development is you're starting with target identification. You've identified a new target in a disease or multiple diseases in a biological process that by hitting that target and either inhibiting it or upregulating it, activating it, you have a change in biological function. And so once you're there, what do you do next? Well, now you've got to figure out, what do you hit it with? Do you hit it with a small molecule? Do you hit it with a biologic? Can you treat it with gene therapy? So what is your modality? Now, how do you screen for that? How do you identify what the right therapeutic is? So you're getting to your lead identification. And so this is where you're kind of then moving forward from, now you've got a lead molecule, you've got a lead biologic, you've got the antibody, and now you're taking it through development candidate profiling. So now you're looking at all the biology around it. And this is where, again, partnering with somebody like West Loop and Deerfield is, they're going to give you the research dollars to continue doing that development candidate while they're doing all the IND enabling work. So you get to do all the mouse studies back in your lab, or at least you get to develop the techniques that you need to do all the mouse studies. And then, okay, now we've developed all these techniques. Now we need to run this through 200 mice. Well, you don't want to run 200 mice in your lab, so they'll go hire a CRO to use the techniques that you've developed. So that's kind of where the partnership works, is that you're developing a lot of the techniques. And that's really important part of the work, because you're the ones with the expertise, you're the ones with the know-how. And then they'll do all the IND enabling studies. This would include all the toxicity, all the safety profiling, all of the formulation work, the scale up that goes into taking something from what you're doing in the lab to being able to sell it on the market. And so, creating the value then to finally get to the market.
And you enter at any stage of this?
Yes. So for the Deerfield projects, people can enter in at any stage. When we initiated this, we thought they were going to be looking really at the early stage. We thought they were looking at target identification because they wanted things that hadn't even been published. They wanted the gems. But what we're finding is that they really want something that you got the gem that nobody else is working on. You've got a novel target or a novel way of hitting that target, but that you probably at least already have identified the modality and that you have at least shown some, probably some biological validation in a mouse model before they really want to be, before they're really going to be interested. You don't have to be all the way to lead optimization, but what we're finding is that the closer you get to lead optimization, unless it's really something that's never been thought of before, that's really where they're looking, is at lead optimization. But they're not looking for somebody who's already gotten 5 papers published to get to lead optimization. So they're looking for somebody who's kind of been saving it all in their lab and now is putting out the big publication at the same time, so that they're kind of getting that first look at it also. So that's why it's been difficult to find the projects, I think, because they want them in just that sweet spot that they're not competing with anybody else. And honestly, what we find at the university is if we get to lead optimization, UIC has been really great at going out and forming relationships with companies. And so when we get things to lead optimization here, we're usually pretty good at getting directly to the license. And so that's where we get into the competition. If it's not, or if Deerfield's not picking it up earlier, we're better off going and partnering it directly with a biotech and pharma who's going to be able to develop, and we don't have to share the cost in the end if they get to the product. So it's a business decision for the UIC.
I wanted to ask you one question. Like you're talking about these therapeutics, right? So for cancer, you have these small molecules, which you use. So supposing somebody says, okay, this small molecule is good for prostate cancer, right? But then some researcher, and so somebody patents that small molecule, and but then some researcher finds, okay, it's not only good for prostate cancer, it's good for, say, lung cancer or some other cancer. Is that a patent discovery or that's not?
It depends on what was already written in the other publications. So typically when people file a patent, what they'll have is they'll have a claim that says, method of using this small, first of all, they'll have a composition of matter claim. So they'll claim the composition no matter what you're using it for. So we're never going to be able to claim the composition again. And then they have additional claims to the method of use. And so they would probably have a specific claim to oncology, to any cancer. And then they would have another claim to pancreatic cancer. And then they might have stuff in their specification, so in the body of the patent, that says, they tested in pancreas, and so they have all the data on pancreatic cancer, but they have nothing on lung cancer. So when they get to the claims and they say all cancers, the examiner says, no, we're not going to give you all cancers. We're only going to give you a claim to pancreatic cancer. Well, now you can come back and try to get a claim and file a patent on lung cancer. Those are often very difficult to get because the other person already said one skilled in the art could figure out that it could be applied to lung cancer. And so, yes, you have to get the data to show it. So they didn't have the enabling data to get the claim, but they've already said that somebody skilled in the art could go and get the data. And so it's that not obvious. So it's really difficult to get those claims unless your data is exceptional. So if it worked 10 times better in lung cancer than pancreatic cancer, now we would go after it in terms of filing the patent and risk that expense, because we have something that's non-obvious because it works even better in lung cancer. Does that make sense?
Yeah.
Or any modifications to compound.
Yes. So if you find that the compound that worked in pancreas needs a small modification and now it works in lung cancer, but it didn't otherwise, now we can get composition and matter claims on that change in the modification. And now we have an even better claim because we have composition claims. So this was an interesting slide to me the first time it was shown to me in terms of how to think about what makes a good therapeutic. And so kind of looking through, and the first thing is genetic evidence. So when you've identified a new target, if you can show that there's a genetic link between that target and the disease, it is 3 times more likely that that therapeutic will make it to market than one that doesn't have a genetic link. So that's what the data has proven out in the last 10 years. So that's an interesting thing from a VC perspective, that they're going to be looking to fund projects that are earlier stage if they have that direct genetic link, because there's a higher chance of probability of success. And so that's kind of where VCs tend to start. So that was an interesting thing to me. I had never thought about having to have that genetic link, and that genetic link can be a direct genetic link. So, like June and I have talked about SMA. Obviously, SMA has a direct genetic link to SMAF, but you can take a lot of other genetic diseases or other diseases that don't have a direct genetic link, but if you knock down that gene is important. So that to them is still genetic evidence that that gene is important. So if you can knock it down and knock out the disease without causing any other harm, well, now you can find a molecule that inhibits that gene or the proteins that are made from that gene, then that's to them at least genetically associated evidence, if not direct genetic evidence. So that's still going to be stronger. So that's kind of the first thing that they're looking at. And if you don't have that genetic link, so a lot of diseases have no genetic link whatsoever in the disease. Then you need to show what they're looking for then, and that's where you have to be further along, because now you have to have some kind of proof of concept that you can put it into an animal model and knocking down or activating that gene or that protein is actually going to cause a biological relevant effect on the animal. And that's much harder to do. So then you're much later along. And then again, you come after they look at the genetic evidence, the next thing they look at is the biology validation target, validated target. So you're looking for a defined mechanism of action. So have you shown exactly how that mechanism of action works? So do you have the biology in your lab? And have you done all the studies to show that that mechanism is important, how that mechanism works, and that you can't circumvent that mechanism with other mechanisms? So yeah, you can knock this down, but when you knock this down, other genes that are active in the system then are going to overcome and compensate for that deficiency. And in the end, you end up with no actual effect on the biology that affects the disease. So that's going to be really well defined in order for VCs to be interested in moving something forward. So again, when you're thinking and designing your experiments in your lab, you've got to be thinking, well, I'm looking at this target and I'm looking at these genes, and here's my molecule that hits that. Well, now you've also got to look at the broader system, and how can you do those studies to look at the broader system to see what effect it has, if you start introducing other than proteins into your even cell assays. What effect do you get? Does it overcome? And so it's kind of, how do you design those studies even at the cell level before you get into animals to see what the biological effect is going to be. And then finally, what they're also looking for is, how can you increase your probability of success? What do you have for biomarkers? What do you have for animal models? How good are the animal models? How many different animal models are out there? Do you have to, are there 3 different animal models that you can use? And between the 3 of them, if they all work, well, now you've truly gotten the effect on the biology you're looking for. Versus, well, we only have this one animal model. It's only been validated for 20% of the patients with the disease. We don't know if other patients that have the disease can truly have this effect, or if this would relate to the biology. Well, then you probably don't have the right animal models, and their VCs are going to be less interested in moving that towards a druggable product. And what we've learned is pretty much what the VCs are interested in is what they think big pharma is interested in. So again, that's what we're kind of thinking about in terms of when we're looking early stage. We're also going to be looking here and saying what assays, what biomarkers, what animal models exist in order to get you to the clinic. Because if we can't get to the clinic, is there really a need to file a patent in the beginning on a therapeutic if we can't get to the end because we can't get the money or we can't get the interest from a licensing partner? And again, the point that Kunjan said about filing a patent is, we want to file the patent because it's incentive to commercialize. And the patent alone is not enough incentive to commercialize if there's a lot of barriers to it. So that's something for in your labs, you think about it, hey, I have something cool, but I know that there's really not a good animal model. Well, maybe part of your lab that needs to be writing that early R01 to get that, to develop that animal model, or to finding that collaborator that can develop that animal model. So something for you to be thinking about in terms of how you're designing your research and how you're designing the studies.
Can I ask you one question? So for example, some of us don't work on the animal models, like we work directly on human beings. Like we just take human beings and do all kind of stuff on, the blood, the tumor, and all that kind of stuff.
Mm-hmm.
So do you need an animal model if you're working on a human being?
Sometimes you do. So I think of, I'm working with a researcher right now who works in RA. And so she has all the blood samples and the synovial fluid samples, which are really good indicators of what might work and what not work in disease. But you still need the animal models, and once you have the drug, to test it in the animal models to see if you actually get the desired effect.
So this is not just for a drug. For example, we are looking on tests like detection of some cancer, right? We want to see, okay, people, we're looking at that. So do we really need an animal? Because there you can't get an animal.
So if you're not developing a therapeutic, then you probably don't need the animal model.
So for those kind of studies, where you're directly studying something in the blood, from biomarker for detection of disease, you don't need an animal model. Only if you're studying a therapeutic, you need an animal model, right? Is that right?
That there might be other reasons you need animal models, but if you're developing a diagnostic,
right,
typically you don't need an animal model, correct? But what diagnostics are a whole nother story, and Kunjan and I can talk to you probably a little bit later about diagnostics. Diagnostics represent a much harder both patent space and commercial space to move through because of the type of work that you do in order to develop the biomarker. One, it's you're typically trying to protect something that's naturally occurring because you're detecting something that's already in the body. So you've got to have a process to detect that, or you're detecting some kind of change that's happening with a process. And so those are much more difficult patents to enforce and to get relevant claims. And two, they typically involve algorithms. So typically when you're developing biomarkers, if it's not one naturally occurring biomarker, if you're developing something that's going to be analysis of several biomarkers, you're using an algorithm to say it's got to have this profile in order to detect several biomarkers at once, so that some are upregulated, some are downregulated in some proportion to each other in order to indicate a disease state. And so we can't file necessarily on that algorithm because that's not patentable either. So then we're filing on the entire process that you go through to do those steps. And if there's something difficult in that process that makes it unique of what you're developing, for example, what Matthew's doing right now with the urine samples that hasn't been done yet, but seems obvious to do if you knew what the biomarkers were, it's still a difficult process. So by perfecting that process, you can patent that. But if you were to try to take routine ways you're doing something in your lab and try to patent that for detection, anybody can overcome that. That's really hard to file a patent on because you can't enforce it. And then the last section of this slide is therapeutic tractability. And so really that's coming down to, do you have the right modality? Do you have the right lead molecule? Do you need to do some formulation work? Do you need to do, can you scale up what you have? And so it's a probability of success of, now that you know it works, can you actually get it to the market? So that's kind of the full thing of what they're looking at. And so, you know, I was working with a faculty who had a really large biologic. He had 100 kilodalton protein. And they got all the way up to here. It was beautiful. They thought they were going to go to the market, and you can't scale up the protein. There was no way to make it in large enough quantities to put it on the market. So they had to go back to the drawing board and figure out how to make a smaller protein or how to make a protein that was a derivative of it. And now you're starting all the way back over to bring it through. So things that if you thought about it earlier, well, can we actually scale this up? Can we make it in a bigger batch than we're doing in our cell studies? Then you probably would have designed it differently in the beginning. So, you know, and again, Dr. Ramaswamy knows very well what we're talking about in terms of looking at the fusion proteins for the vaccine in terms of when you go to scale it up, you know, what effect is that having? Can you actually get here? What is the probability of success of doing that? And so a lot of those kind of questions that we were looking at in those previous slides are kind of the should we do it? You know, does it have a clear mechanistic hypothesis? What is the value proposition? To then the can we do it? So even if it looks like it's a right target, it looks like the biology, you know, that hitting that target is a perfect in the biology, you know, do we have the right tools and reagents to actually satisfy the FDA requirements? Can we actually scale it up and get to the market? Do we have a biomarker to use in clinical trials so that we have a way to get to patients in a trial with 200 patients? Because again, if you need to do your clinical trial in 20,000 patients because there isn't a good biomarker, is pharma really going to pick it up and run that 20,000 patient clinical trial? Or are they going to be like, no? So, you know, or is there some way of stratifying your patients with a biomarker or some way of measuring the disease onset? So these are kind of the should we and can we do it questions that come from that previous slide. I included this really for you guys to have later, but this is just going into some of the questions that touch on the biological rationale in terms of when you're thinking about early research and questions that you can be asking yourself so that if your research was to advance to a therapeutic, have you answered these questions and can you actually advance to a therapeutic? Because if you can't go through a lot of the things on this slide, if they apply to your research, even though it's really great research and it can get you a lot of ROI funding, whatever that therapeutic pathway is probably not going to get to the market. So things to just keep in mind in terms of the validation along the way. And again, this is about the therapeutic tractability. So this is about the stuff at the end of, you know, we have a lot of faculty on campus who are developing peptides. And peptides are really tricky to get to the market because your peptides can be easily digested by enzymes. So how are you delivering? Do you have a delivery system? You know, are you attaching a carrier that you need for your studies in mice that you can't bring into humans So what happens when you take that carrier off? Does it still have all the same effects? Or can you leave that carrier on, but that carrier has toxicity signals, so you have to take the carrier off? So things like that. So those are the kind of questions that, again, things to keep in mind when you're doing your early research is the safety, the pharmacokinetics, how long does it last in the body? You need something that needs to be delivered systemically, and you need it for a chronic condition. You want it to last in the body for 24 hours or more, or if you can do slow release and you only have to inject it once a month, that's better than something that you're giving chronically that has to be taken every 4 hours. So how does patient compliance even come into this? So all of this factors into therapeutic tractability in the end. So just to kind of keep in mind that the drug development is not just about, I have a great molecule that hits my target. It's about how do you get it to the end. And then this is just kind of a timeline because we love our timeline. And so what we're seeing is basically it is anywhere from 9 to 12 months from when I start having a conversation with you before there might be an agreement. And when I'm saying 9 to 12 months, that's probably generous. We're probably seeing closer to a year for Deerfield to work through their process because they're taking into account all of this information. So they're starting with what you have in your lab, what molecules you have, what screens you have, what assays you have, what capabilities your lab has, what capabilities we might need from a CRO, what CROs can do the work, what is all the work that needs to get done all the way to be able to put that molecule into a patient the first time. So they're looking at all the work that needs to get done and evaluating everything. And then we're writing that full proposal to do what's probably going to account for 3 to 5 years worth of work with them. So that can take some time. So it's a pretty lengthy process, but we're hoping to get 2 projects in by the end of this year and fund it using this mechanism. So, and like I said, a typical timeline for getting a therapeutic to market is probably 15 to 20 years, which happens to line up nicely with our patent timeline of 20 years. So anything that we can do to accelerate it so that we're in the clinic in less time, knowing that the clinic is going to take anywhere from 5 to 7 years. So if you can take this time down to 4 to 6 years, which is what Deerfield is hoping to do, and then you add 5 to 7 years on your clinic, you still have 8 years left of your patent life, which means 8 years of exclusivity on the market, which means more revenues back to the university and to the faculty that created the technology. So that's what they're kind of hoping. And I put both of these timelines up really just to show you the difference between a small molecule and for here an antibody. And any biologic is probably going to be closer to the antibody in terms of your steps are almost the same. They vary slightly, but the timelines between them can change depending on whether you're looking at a small molecule or a biologic. One of the advantages that I do have in working with Deerfield is that they have a huge network of partners. And so we've been able to tap into these. So we're currently looking at a monoclonal antibody project that's working with the Plexus that has a unique model for developing monoclonal antibodies. We do have access to Schrodinger, which is a computational program. And so I get free tokens. So anybody who uses Schrodinger, if you know what that is and you need more tokens, come and ask me. I do have a way to access those for you. And then again, what we've gained is really doors being open for us. So Deerfield, being a huge venture capital, have funded a lot of small biotechs that are now medium-sized biotechs. And so when we do have science in certain areas, even if they're not interested in funding it, they say, but we had a company that we funded 5 years ago that's now got an IPO and they're in this space, they're probably looking for their next asset. And so they make the introduction. And so we've gotten a lot of introductions to companies from Deerfield in terms of opening doors to let researchers say, hey, I'm doing this in my lab. And that's opened a couple of licensing opportunities and potential research collaboration. So we're tapping into that network. And this is just kind of to show you what I've been doing in the last 2 years. So I've probably talked to over 200 faculty since I've been taking on this role, and did a deep dive on probably about 80 faculty projects where we've actually dove into kind of what is the target, what is the disease, what other diseases can we go into, is there a way to take this to the market, and what are the pitfalls in terms of working with them in different aspects of their projects. And so we've gotten from the original 39, we did 6 deep dives, and from that I think we've gotten now 4 of those or 2 of those left, 4 firm no's, and the other 2 are probably still here in the ongoing discussions because we're finding internal funding for those. So we found ways to keep those projects going and moving forward. I'm continuing to have conversations with Deerfield, and we're hoping that eventually one of those projects will go in to funding. I put formally 10 projects in, 6 through proposal concept sheets and 3 more through LOIs to formally be evaluated by Deerfield, where we've gotten amazing feedback back from them in terms of kind of what the pitfalls are of moving those forward in therapeutics and what kind of our next key studies we need to do in order to advance those projects. And so those have moved then into these continuing ongoing discussions. So that's kind of where I sit in terms of looking at. Right now, I've got about 4 faculty that I'm working with, trying to put in a proposal concept sheet or a letter of intent to see if we can get funding through that. June and I started talking 2 months ago, and we're hoping to put one of her projects in. And I talked to Les, I guess last month for the first time. So hopefully we'll be putting that project in maybe the end of the summer. So I'm excited to talk to anybody who I haven't already talked to. If you have anything in the space, please come and introduce yourself. And if we can't talk today, we can talk via Zoom anytime.
Thank you.
So Maderia, thank you so much. So I have one quick question. I think these are very useful, this information. One quick question is, I know that previously, in the UI center, the Discovery Center, they have a library of these kind of drugs. Is that still available to people to test?
Yeah, so actually, I don't know if you guys heard this, I don't know if this recorded, support. So the UI Center did recently put an offer out, and my understanding is somebody accepted.
Oh, okay.
So they do have a new director coming into the UI Center, which will be fabulous then if he joins in the fall.
Okay.
I think that a lot of the programs that will get revamped.
Okay.
So they do have, they still have 3 people that are working for UI Center. I think they've hired 2 chemists, right, Nelson?
Yeah.
So they have the chemists and then the 3 people who can run the assays and do the assay development. And then they have, I think it's 2 different libraries of compounds.
Yeah, they got ChemDiv, I think it's one of them.
ChemDiv, which is like 20,000 compounds, and then they have another one that's
Right.
about 20,000
Yeah,
compounds.
yeah.
So they have access to 2 different libraries to run.
Thank you.
So this West Loop and Discovery EPI, there is another like Discovery Partnership.
Discover Partners
Is it
Institute.
like how the connections, different way. So
Okay, so with West Loop, I sit under the Vice Chancellor for Innovation, but I still sit within OTM, so I still work closely with OTM. So we look at West Loop as one potential way to take research out of UIC and partner it to get developed. DPI is basically a state funding mechanism, and so they've begun to hire and fund projects through DPI. And I think they have I think they have about 20 employees now that actually work for DPI. So they're kind of building up their infrastructure. And they have some faculty that have been funded specifically through that mechanism, where they're starting to have some inventions. And then because they're still UIC faculty or UIUC faculty, they still work within our mechanisms as far as filing patents. And hopefully they'll be announcing more information about DPI in the next year, because I think they're going to be launching a lot more funding mechanisms for that. All right, I guess Shinja and I will be here through lunch, and Nelson will be back there. So feel free to come and chat.
Do you want to just double check whether there's any question on the chat?
yeah.
Okay.
For the Zoom participants, if you have any questions, of course you can. But on behalf of Office of Research and our campus, we want to thank you, coming over here and explaining a lot of what we are going to do or focusing in Chicago. The way in which we planned is like we have a lunch box is here, so we are available. I think before next meeting 1:30, you have some